So here's that animation, and then we'll go over the steps one more time and then proceed to the next section.
Spore-forming bacteria grow as vegetative cells and divide by binary fission when nutrients are available and environmental conditions are not adverse. When nutrients are depleted or conditions become adverse, spore formation is initiated. The DNA condenses and aligns itself in the center of the cell. The vegetative cell is now referred to as the mother cell. Next, the DNA divides into two complete copies, and the mother cell membrane invaginates to form the developing forespore. The mother cell membrane continues to grow and engulfs the developing spore. The developing spore is now surrounded by two membrane layers. Next, peptidoglycan is laid down between the two membranes of the developing spore to form the cortex. Dipicolenic acid is formed inside the developing spore, and calcium enters from the outside. As calcium enters the spore, water is removed. A protein coat forms exterior to the cortex, and the spore becomes mature. Some spores form an additional layer called the exosporium. A mature spore is resistant to environmental conditions. Finally, lytic enzymes destroy the mother cell and the mature spore is released.
So here are the steps of sporogenesis or sporulation. The chromosome divides, you have DNA replication. Then the cell membrane divides asymmetrically, divides the cytoplasm asymmetrically. So it invaginates asymmetrically. So one side is bigger than the other. The bigger one is called the sporangium, and the smaller one is called the forespore. So the sporangium engulfs the forespore, the bigger one engulfs the smaller compartment. And then the peptidoglycan is added to the space between the two membranes and of the forespore, and it is now called the cortex. Then a spore coat or protein coat is formed around the cortex, and so it's now called the mature spore. And the mature spore is released as the vegetative cell, the mother cell, dies. So that is those actually 6 steps. And what I'm going to have you do, I will give you the first and the last step, or 2 of the steps, and you have to put the other steps in order and be able to know what happens before the next steps. You should be able to logically kind of figure it out, because if the forespore is formed, then, then that can't be before the sporangium engulfs the forespore. But anyway, that's how I asked the question on the test, is for you to put these steps in order. So I would suggest making little flashcards and putting the steps in order and practice putting them in order. So germination is when they get enough water, enough nutrients, so when favorable conditions arise, germination agent will stimulate the formation of a hydrolytic enzyme that breaks down the cortex, because remember, this is very rigid, so it has to be broken down, and then the core rehydrates and takes up the nutrients, and the bacterium grows out of the endospore coat. And once that's initiated, the time for this is about an hour and a half. So it only takes about an hour and a half once they get into favorable conditions. So some of the organisms that produce this, they have the genus Bacillus or Clostridium. So anthrax, which we've heard about, which causes a pretty severe respiratory illness, very deadly. Clostridium tetani, which causes tetanus, lockjaw. Clostridium perfringens causes gas gangrene, and Clostridium botulinum causes botulism, which is also what they use. That's the toxin from this bacteria is used in Botox treatments. So those are some of the diseases related to the spore persistence. But Bacillus is a very aerobic bacteria, and Clostridium are anaerobic bacteria. So different conditions will cause them to become. So the medical significance of endospores, They are intruders. They're constant intruders where sterility and cleanliness are important. So in a hospital setting, in wound treatment, in culture media, you know, here in the lab, we want to make sure that we do not have any endospore-forming bacteria, because the regular methods of cleaning and disinfecting are no longer, they're not effective, because disinfection, by definition, does not kill endospores. And boiling water, soaps, and disinfectants, you know, using disinfectants, all of those are disinfection. So hospitals, food canning industry, we want to keep out the endospores, because just boiling is not going to kill the endospores. So here's our concept check for section 4.4. True or false, plasmids are vital to the metabolism of bacterial cells. How are bacterial ribosomes different from eukaryotic ribosomes? List and describe the steps of endospore formation and germination. Name the three diseases caused by endospore-forming bacteria. So plasmids are vital to metabolism of bacteria cells. That is false. They are not required. They're kind of extra credit. You know, my extra credit analogy. So they're not vital, but they do help bacteria survive in situations where they normally would not be able to survive, because they have some beneficial trait that they give to the bacteria. So how are bacterial ribosomes different than eukaryotic ribosomes? Bacterial ribosomes are 70S with a 50 and 30S subunit. The eukaryotic ribosomes are 80S, and they have a 60 and a 40S subunit. So they're different sizes, and that means that they're also different shapes. And that's important because, and I didn't mention this earlier, but the shape and the size of the ribosomes is being different is important, because we can use antibiotics that will inhibit the ribosome subunits of bacteria, and they won't inhibit our ribosomes. Our ribosomes will be not affected. And those antibiotics can't get into the mitochondria, so they're not going to affect mitochondria metabolism either. So they will be able to kill the bacteria without killing or hurting us. That's a selective toxicity. So that's a really important difference between eukaryotes and prokaryotes that we use to treat infections. And so then the list of the steps of endospore formation, you have the replication of the DNA, the asymmetrical division of the cytoplasm into a forespore and a sporangium, the sporangium engulfing the forespore, peptidoglycan laid down between the layers of the forespore, and then a coat, a protein coat forming around the outside of the endospore or the cortex, and making the mature spore, the mature endospore, and then the release of the endospore. So those are the steps. Name the diseases caused by endospore-forming bacteria. We said anthrax and tetanus and gangrene and botulism. Those are just a few that we can name. So that is the end of section 4.